Hello, my name is Jeffrey Campbell, and welcome to my presentation on behavioral epigenetics, or an overview of genetic memory. Epigenetics in general can be defined as the way that the cell regulates the structure of its genetic material. This can serve many different functions within the cell. However, behavioral epigenetics refers specifically to the types of modifications made to the genetic material in order to promote more optimal behavior in the macroorganism. This can be done through a variety of methods. However, the two best understood and most well documented ones are methylation and histone modification. Histones are small barrel-like structures that DNA is wrapped around to form a genetic structure known as chromatin. By modifying the affinity that these histones have for binding to each other, the cell can regulate the density of the genes in its nucleus. This can have many effects. However, its primary effect for epigenetics is to regulate gene expression. When histones are very closely associated due to binding, cellular machinery cannot bind to DNA and effectively cannot transcribe any gene present in the tightly bound chromatin complex. However, when modifications are made to the histones, and this could come in many forms from many places, that essentially reduce their affinity to bind to each other, the chromatin becomes far less compact, and cellular machinery can bind to it and transcribe the genes within it. Methylation is the addition of a methyl group, or one carbon and three hydrogens, to either a cysteine or a guanine amino acid. This can have a wide variety of effects depending on where it occurs. If it occurs in a promoter region for a gene, it will make that promoter unreadable from the perspective of cellular machinery, effectively silencing and ending all transcription of that gene. However, this can also have the effect of recruiting proteins to the area of methylation that would interact with cellular machinery in order to increase or decrease the expression of a particular gene. Or it can exclude other proteins that would otherwise be binding to a particular sequence and have an impact, positive or negative, on gene expression. However, generally speaking, it is most common for methylation in DNA to indicate a reduction in expression of genes. How and why these modifications occur is still an area of debate, but some studies have found direct examples of an animal's environment affecting modifications to its epigenome. One 2012 study investigating the effect of maternal grooming on the stress responses of mice found that an increase in maternal grooming resulted in an increase in the methylation of the estrogen receptor promoter region of those mouse pups. This region is responsible for creating the proteins that allow mice to be affected by the hormone estrogen, and its methylation would explain why mice that were groomed more as infants had a very reduced response to stress, as estrogen is one of the hormones most responsible for increasing stress responses, especially in mice. The heritability of epigenetic modifications is not fully understood, but has been clearly seen to occur. It is now believed to be responsible for many rapidly emerging generational traits that occur too quickly to be the result of random mutagenesis to the nucleotide sequence. A 2012 study found that an altered stress response in mice could be inherited by the mice's descendants, even 2 or 3 generations down. Descendants of mice that were kept in a high-stress environment were on average 25 grams heavier than the average non-stressed line mouse, and showed 10% more fearful behavior in the form of wall hugging and hiding when introduced into a novel environment. While it may seem pointless to understand the mouse epigenome, many of the mechanisms that control epigenetics appear to be conserved, at least partially, between species. In 1988, while investigating similar methylation binding motifs in several species of vertebrate, namely cow, human, and rat, researchers managed to isolate a protein that was responsible for DNA methylation and believed to be conserved across all species. When introduced to a piece of control DNA, all of the proteins from each of the species, cow, human, and rat, bound and methylated the DNA in the same way and with the same sequence specificity. However, it was unable to bind DNA that was provided from a mosquito and other non-vertebrate species. Taking into consideration how specific the changes in behavior epigenetics can cause, the fact that they can happen at any point in life, and the magnitude of change that they can create, it seems more than possible that epigenetics may one day play a large role in improving both our physical and psychological well-being. But how and when has yet to be seen. Thank you for watching, and if you have any questions or comments, I'll be sure to get around to addressing as many as I can. Bye.